Good morning, students. So our chapter excretory product and their elimination is going on, and this is the part 3. Now today we'll study about urine formation. Now a term is there, glomerular filtration rate. First of all, we'll study what does it mean. The glomerular filtration rate, the quantity of glomerular filtrate formed each minute in all the nephrons of both kidneys is called glomerular filtration rate. In the normal person, the glomerular filtration rate is 125 ml per minute. It means that if we multiply 125 into 60, then it will become per hour, and we'll multiply it with 24 hours, then it will come out per day. It means that 125 ml per minute means 180 liters per day. Now we'll study how 180 liters per day converts into 1 or 2 liters urine that we excrete out. So in the process of urine formation, there are three main processes: glomerular filtration, reabsorption, and tubular secretion. Now we'll study one by one. First of all is glomerular filtration. What does it mean? The first step in urine formation is the filtration of blood, which is carried out by glomerulus and is called glomerular filtration. On an average, 1100 to 1200 ml of blood is filtered by the kidney per minute, which constitute roughly 1/5 of the blood pumped out by each ventricle of the heart in a minute. The glomerular capillary blood pressure causes filtration of blood through three layers. Now we all know that there is one first layer is endothelium of glomerular blood vessel, and the second layer is the epithelium of Bowman's capsule, and in between these two there is a basement membrane present. So the blood which is passing through, which is getting filtered, has to pass through three layers. First one is endothelium of glomerular blood vessel, second one is basement membrane present in between them, and the third one is called the epithelium of Bowman's capsule. So the epithelial cells of Bowman's capsule, called podocytes, are arranged in an intrinsic manner so as to leave some minute gaps between them. These are called slit or slit pores. Blood is filtered so finely through these membranes that almost all the constituents of the plasma except the proteins pass on to the lumen of the Bowman's capsule, and therefore it is called as a ultrafiltration. So I just told you about glomerular filtration rate. The amount of filtrate formed by kidney per minute is called glomerular filtration rate. The kidneys have built-in mechanism for the regulation of glomerular filtration rate. One such efficient mechanism is carried out by juxtaglomerular apparatus. What does it mean? Juxtaglomerular apparatus is the sensitive region formed by the cellular modification in the distal convoluted tubule and the afferent arteriole at the location of their contact. Means where distal convoluted tubule meet with the afferent arteriole, that particular place is called juxtaglomerular apparatus. A fall in the juxtaglomerular rate can activate the juxtaglomerular cells to release renin, R-E-N-I-N, which can stimulate the glomerular blood flow and thereby the glomerular filtration rate back to normal. Now you must notice that the spelling of this hormone is R-E-N-I-N, that is in the chapter excretory products and their elimination. And one more renin we have studied in digestive system, RENNIN, which is used for conversion of casein protein present in the milk, to simplify casein protein present in the milk. So you have to understand very well the difference between renin, RENNIN, and RENIN. It's clear? Now next we'll study what tubular reabsorption. Now, tubular reabsorption means that absorption of selected material takes place from the filtrate into the blood of the peritubular capillaries of vasa recta. It is termed the tubular reabsorption. Tubular reabsorption involves both passive and active transport across the tubular epithelium. As already I have told you, the glomerular filtration in the filtrate in the Bowman's capsule resembles blood plasma in composition except for plasma proteins and fats. Therefore, it is almost isotonic to the plasma. Now, you must notice that in comparison to the volume of filtrate formed per day, that is I told you just in the previous slide, 180 liters per day. With that of the urine released, how much urine is released? It's approximately 1.5 to 2 liters. Suggests that nearly 99% of the filtrate has to be reabsorbed by the renal tubules. So this process is reabsorption. So we all know that some substances such as glucose, amino acids are reabsorbed actively into the blood by active transport, and therefore they don't appear in the urine. These substances are called the high threshold substances. So the renal threshold of glucose is about 180 milligram per 100 ml of blood. When this value is exceeded, glucose begins to appear in the urine. And then we call the person is diabetic now. If urine is contains glucose, some amount of glucose, then the person is called diabetic. Now, now the third process which happens in the urine formation is tubular secretion. The cells of renal tubule not only remove substances from the filtrate by the process of reabsorption and send them to the blood capillaries, peritubular capillaries, but also excrete additional waste from the bloodstream into the filtrate by the process of secretion. Thus, tubular secretion is the opposite of tubular reabsorption. Just like creatinine, pigments, drugs including penicillin are actively secreted into the filtrate in the proximal convoluted tubule from the interstitial fluid. Hydrogen ions and ammonia, potassium ions are also secreted into the proximal convoluted tubules. Urea enters the filtrate by diffusion in the thin segments of ascending limb of Henle. Now, potassium, hydrogen ions, ammonia, as I have told earlier, bicarbonate ions are secreted by active transport into the filtrate in the distal convoluted tubule. So, maximum hydrogen secretion occurs in the proximal convoluted tubule. Removal of hydrogen ions and ammonia from the blood in the proximal convoluted tubule and distal convoluted tubule helps to maintain the pH of the blood between 6 to 8. pH of blood is usually 7.4. You all know already. So tubular secretion plays a minor role in the function of human kidneys, but in animals like marine fishes, desert amphibians, and whose nephrons don't possess developed glomeruli, their urine is found mainly by tubular secretion of urea, creatinine, mineral ions. Kidneys excrete about 1.5 liters of urine in a day. This slide is only for your revision to revise the difference between tubular reabsorption and tubular secretion. Tubular reabsorption, once again I am telling you, it is absorption of selected materials from the nephric filtrate into the blood of peritubular blood capillaries, and tubular secretion is it is a removal of selected materials from the blood of the peritubular blood capillaries into the nephric filtrate. So you must understand the difference between both.